Hello, everyone. Welcome back to another video. We're going to do lecture on metabolism and cellular energy. Before we get started, please like and subscribe to the channel if you have not done so. Also, look at our Patreon, patreon.com/twistedscience for more material. So we're talking about cell metabolism today. Metabolic processes are going to be everything that creates and consumes energy in the cell. So this is our series of processes that are going to allow for cellular metabolism to take place, right? Each living cell and every organism has metabolic needs and metabolic processes. So the nutritional use of metabolism is going to refer to the chemical processes in our bodies that convert food into energy. On each level, whether it be the cellular level or all the way up to the largest individual person or animal, we're going to have respiration that takes place and metabolism that takes place in each individual cell. So there are going to be steps of cellular respiration that occur in both prokaryotes and eukaryotes. In a eukaryotic cell, we are going to have four main steps of respiration. Okay, cellular respiration, or process of utilizing energy, is going to be comprised of glycolysis, pyruvate oxidation, citric acid or Krebs cycle, and oxidative phosphorylation. Now, this respiration is for the main purpose of producing ATP. Our main cellular energy source is ATP that we're going to get by utilizing breaking down of bonds such as glucose or carbohydrates. So our main reactants that are going to go into cellular energy for the most part is going to be glucose and oxygen. The main products that are going to come out of cellular metabolism are going to be carbon dioxide, water, and ATP. Now, this is for metabolism in an animal, right? If we're talking about plants, it is going to be from the opposite. So it's going to be oxygen as a product instead of carbon dioxide. So looking at this, all right, photosynthesis in cells is going to be a metabolic pathway utilizing the sun's rays and UV radiation. All right, plants and algae, cyanobacteria, these all use photosynthesis. Those are going to be light-dependent reactions. This is producing energy through what is known as the Calvin cycle, which takes place here, does not take place in animals, okay? The main reactants here are going to be the light energy that we get from UV radiation, carbon dioxide taken in from the environment, and water. The main products are going to be the opposite of what we see in animals. Here, the products are glucose and oxygen, whereas the products in animal cellular respiration are going to be carbon dioxide and water. For prokaryotic metabolism is variable. It depends on which kind of energy substrate they're going to utilize most often. So in terms of metabolic pathways, we're going to have what are known as heterotrophic, autotrophic, phototropic, or chemotrophic reactions. The type of metabolism that a prokaryote is going to be using is basically indicative of where it lives and what it needs to do in order to survive in its environment. We also have a large role that prokaryotes play in ecology, right? Human health and diseases. Our example here, right? There are a lot of prokaryotes that are not able to tolerate oxygen well. They are anaerobic. They utilize anaerobic metabolism. One of this is botulinum toxin, okay? Botulinum is going to be a prokaryote that's going to cause muscle paralysis if ingested and taken in humans. It causes botulism because it goes well in areas without any oxygen. All right, so our metabolic processes are going to take less time than they normally would in other circumstances because of enzymes. Enzymes are going to be what catalyzes or speeds up chemical reactions. Most of our biochemical reactions are going to rely on enzymes to be efficient, right? We need this for cellular metabolism. The two most common energy sources for cell metabolism are glucose and light energy, right? but we still need enzymes in these pathways to get enough energy out to fuel all of our cellular activity. Most enzymes in cells are going to be proteins and lower the activation energy for chemical processes to begin. In eukaryotes, cellular respiration is going to primarily occur in the mitochondria. This is going to be an oxidative aerobic function, right? So we depend heavily on oxygen to allow us to create cellular energy. We start out with the anaerobic part of it, which is happening in the cytosol of the cell, known as glycolysis. Glycolysis is taking the initial glucose molecule and breaking it down with or without oxygen. It's going to split that carbon into 3 carbon units. So we split it into a 3-carbon pyruvate molecule. Also, during glycolysis, we do make 1 ATP and convert NAD to NADH. After we have made our pyruvates and our NADH, we are then going to be converting pyruvate, and this is going to be known as pyruvate oxidation. This is where pyruvate is going to enter into the mitochondrial matrix and become coenzyme A or acetyl-CoA. This step has to have oxygen involved. If we don't have oxygen here, then it's not going to happen, right? This is going to release carbon dioxide and make more NADH. After we have made acetyl-CoA, that acetyl-CoA is going to move into what is called the Krebs cycle, or the citric acid cycle. It's going to combine with a leftover substrate known as oxaloacetate, and this makes citrate. Citrate is going to be the product that enters into the cycle, or enters into the steps of the reactions. So through reactions, we have 10 reactions that happen here. It is going to finish up making carbon dioxide and NADH. The cycle is always going to produce FADH2 and ATP as well. Moving on from there, once we've created these products, the NADH and FADH2s that have been made are going to move on into oxidative phosphorylation, which is happening in the matrix of the mitochondria. So this is where we get the most energy. This is where we need oxygen. This is called the electron transport chain. The NADH and FADH2 that have been produced in the previous steps of metabolism are going to donate electrons to this chain, and they're going to be converted back into NAD plus and FAD. The electrons are able to move down the 4 enzyme complexes within the electron transport chain, producing ATP as they go along. The majority of our ATP production is happening here, right? About 80% of our ATP production comes from this step. In photosynthesis, we have this occur in plant cells, some algae and bacteria as well. These are going to require what are known as chloroplasts. So chloroplasts are going to work similar to mitochondria. However, here, instead of utilizing glucose derivatives, we are going to be utilizing light energy. Chloroplasts are going to contain a protein pigment known as chlorophyll, which is going to be why most plants are going to appear greenish in color. The end product is going to be glucose or sugar along with oxygen. This is where we have the light-dependent reactions, right? So during those light-dependent reactions, inside of the chloroplast, there is what is known as a thylakoid membrane. The thylakoid membrane is going to contain the chlorophyll pigments, which absorb light energy. From this light radiation, we get ATP, a protein known as NADPH, and water. These byproducts are going to enter into the Calvin cycle, which is in the stroma of the chloroplast. The Calvin cycle is going to produce ATP, NADPH, and also G3P, which eventually becomes glucose. There are 2 different types of chlorophyll that are the most common. The third one, chlorophyll C, is a little bit less common, but still of note. So we have chlorophyll A, B, and C. Most plants have chlorophyll A. Chlorophyll A is going to absorb blue and red light wavelengths. Some plants and green algae have chlorophyll B, and chlorophyll C exists in select dinoflagellates. Unlike with animals and humans, prokaryotes are going to vary in their need for oxygen. So we do have those that can exist in aerobic and anaerobic um, environments. Some of them are known as facultative anaerobes, meaning that they're able to exist in both aerobic and anaerobic capacities. So some can exist, some can't, right? Some prokaryotes can switch between the two types, and that is why they are known as facultative. Prokaryotes that depend on oxygen are known as obligate anaerobes, i.e. they are obligated to use oxygen. Prokaryotes that cannot are known as obligate anaerobes. The ones that switch, right, are known as facultative anaerobes. Next is the lactic acid fermentation. Lactic acid fermentation is going to be a byproduct of anaerobic activity. So when we do not have oxygen present, usually lactic acid is going to be a byproduct of this metabolism. So muscle cells are going to be able to do lactic acid fermentation in the human body. We also see that bacteria are going to produce lactic acid to a large degree during anaerobic environments. During this process, the cells are going to make ATP without oxygen. The process is going to turn the pyruvate made in glycolysis into lactic acid. We have many applications in the real world for this, right? Such as with yogurt, ethanol production. All right, these things are big products that we can make from fermentation reactions. For example, the Lactobacillus bulgaricus helps produce yogurt. That is a bacteria. Bacteria are able to ferment lactose, right, and make lactic acid. The lactose is able to make the milk clot, and that is how we turn it into yogurt. All right, so we can characterize prokaryotes by their metabolic type. The main types are going to be heterotrophic, autotrophic, photo, and chemotrophic. Heterotrophic prokaryotes are going to use organic compounds from other organisms to obtain carbon. So this is similar to human beings eating animal products in order to get energy. Here, we are going to have the usage of other organic compounds to get energy. Autotrophic prokaryotes use the carbon dioxide from the environment as their source. Many are able to use photosynthesis as well. Phototropic prokaryotes, as the name would suggest, use photo or light energy, so they are able to use energy from light. Right. Chemotrophic prokaryotes get their energy from chemical compounds that they break down. Two variations of reactions, catabolic versus anabolic. Metabolic pathways are going to be divided into these two categories. Anabolic is going to be building up. It's going to be a reaction that is going to put something together and make a product. Catabolic is going to break things down. So anabolic is going to require energy to build larger molecules from smaller ones. Catabolic is going to break down or release energy from the breakage of large molecules into smaller ones. Photosynthesis is going to be an anabolic process that we see in plant cells, whereas the cellular respiration that we normally see in animal and human cells is going to be a catabolic process. Eukaryotic and prokaryotic cells both are going to rely on cellular respiration and metabolism to survive. Cellular respiration and photosynthesis are the most common pathways that we see in cells. Some more unique things that we see, right? If we get into the world of prokaryotes, we are able to see some variations on this, right? We have a diverse metabolic pathway that exists here. Some of them are able to obtain fixed carbon, right, and energy. Some species are going to consume organic material like dead or decaying plants and animals. Others are going to live off of inorganic compounds like rocks and sediment, right? So dividing our prokaryotes here into 4 different categories. Photoautotroph is going to use light energy. Carbon source is carbon dioxide. Photoheterotroph is going to use light energy and get a carbon source from organic compounds. Chemoautotroph is going to use chemical compounds for energy, and it's going to get its carbon from carbon dioxide. Chemoheterotroph is going to get it from chemical compounds, and the carbon source is organic compounds. Oxygen usage is going to be the primary consideration here for classification. So prokaryotes that need it are going to be the obligate aerobes. Humans, as we know, are obligate aerobes. Prokaryotes that aren't able to tolerate oxygen are usually only able to perform anaerobic metabolism. Facultative anaerobes are going to use oxygen if it's present. If it's not, then they can use an anaerobic metabolism. Examples here are bacteria that cause staph and strep infections. These are facultative anaerobes. All right, additional metabolic pathways. We have sulfur and nitrogen metabolism. Sulfur metabolizing prokaryotes are going to form the basis of many food chains that exist in deep sea habitats. We have sulfurous vents that exist in the deep sea habitats that are facultative, or sorry, are sulfur metabolizing prokaryotes can potentially use for energy. Sulfur metabolizers are going to support the entire community down there, right? Nitrogen metabolizing prokaryotes are going to be able to use nitrogen in the atmosphere as well as in the organic compounds. So nitrogen fixers, nitrifiers, and denitrifiers. They play key roles in the nitrogen cycle by converting nitrogen compounds from one chemical form to another. So nitrogen fixing prokaryotes are going to be able to convert or fix atmospheric nitrogen into ammonia. Plants and other organisms are able to incorporate ammonia into organic molecules. Plants are able to house and feed bacteria in structures called root nodules that exist in the ground. So the bacteria that are able to fix nitrogen can provide nitrogen then to the roots of the plant. Other prokaryotes in soil, known as nitrifying bacteria, are able to convert ammonia into other types. Denitrifying prokaryotes are going to do the reverse of this. They're going to turn nitrates into gases, and they're going to denitrify these components. So here's our nitrogen fixing, right? We see atmospheric nitrogen here. It's going to circulate around. The nitrogen fixing prokaryotes are going to convert the atmospheric nitrogen into ammonia. Nitrogen nitrifying prokaryotes convert the ammonia into nitrites and nitrates. Denitrifying prokaryotes are able to convert nitrates back into atmospheric nitrogen. All right, next is the carbon cycle. Prokaryotes are going to be the most important constituent for the carbon cycle that we have. So photosynthetic prokaryotes use light energy to remove carbon dioxide from the atmosphere. Right? This is the same basic process as what we see in photosynthetic plants. Prokaryotic decomposers are going to move carbon in the opposite direction. They're going to break down organic material and return carbon dioxide to the atmosphere via their cellular respiration. So the decomposition is going to release a variety of other elements and inorganic molecules. So here we take atmospheric carbon in the form of carbon dioxide. Plants are able to fix this carbon dioxide during their metabolic reactions. Photosynthetic prokaryotes are able to fix the carbon dioxide as well. Decomposing prokaryotes are able to convert fixed carbon back into carbon dioxide, which then is passed into the atmosphere once more. All right, some of the specifics of cellular metabolism as far as glucose is concerned. Breaking down glucose is what we mainly do in human cells and animal cells to get energy. So most of the ones in our body get energy from glucose, chemical compound here, C6H12O6. The overall reaction of this process is taking 6 oxygens, right, 1 for every carbon that exists in the glucose molecule, and reacting that to form the products of 6 carbon dioxides, 6 water molecules, and cellular energy in the form of ATP. Our energy there in the form of ATP, adenosine triphosphate, is our standardized energy transfer, right? It's known as the energy currency of the cell. Plants are going to be unique in they're able to build up glucose by using photosynthetic reactions. So the photosynthetic reaction is going to be the cellular respiration pathway, but reversed. Here we are going to take the carbon dioxide from the atmosphere, combine it with water and energy, and form glucose from that. So we get the reverse reaction. Instead of having glucose and oxygen as the reactants, here we have them as the products. These chemical pathways don't take place automatically. We need enzymatic help. So each one of these steps in the reaction is going to be catalyzed by enzymatic control. Enzymes are able to control the rate and strength of all biochemical reactions. So when we group prokaryotes together based on their metabolism, we are going to group them into these specific classifications. Heterotropic are going to be ones that use organic compounds from other organisms. Autotropic are going to be ones that are going to use carbon dioxide as their only source of carbon, right? Many of these are photosynthetic. Some of them get carbon dioxide from the atmosphere. The process of capturing inorganic carbon and converting it to sugar molecules is known as carbon fixation. We also see energy metabolism classification. Phototropic, ones that capture light and convert it to chemical energy. Chemotropic, ones that break down organic or inorganic compounds to supply energy for the cell. Some of our chemotropic organisms are able to use their own energy supplying molecules, which makes them chemoheterotrophs. To classify something as organic, we're going to have to be able to put it into an organic category. So we have to make it in the realm of living things or in the realm of a product of a living constituent. So photoheterotrophs, or organisms that convert light energy to chemical energy, but they get their carbon from organic sources. So they're able to break down organic sources to get carbon, such as soil and plants. All right, examples of this are going to be purple and green non-sulfur bacteria. Chemoheterotrophs are going to be ones that get their energy from chemical breakdown, but they get their carbon from organic sources. Okay, they must consume organic building blocks that they're unable to make themselves. Most are going to get their energy from sugar, such as glucose. Photoautotrophs, going to capture light energy and use carbon dioxide. We have many photoautotrophic prokaryotes. Photoautotrophic prokaryotes are going to use similar compounds to those that trap plants that trap light energy. Chemoautotrophs break down inorganic molecules to supply energy. Chemoautotrophs include prokaryotes that break down hydrogen sulfide and ammonia. Nitrosomonas is a species of soil bacterium that oxidizes ammonia and turns it into nitrite. So this is a nitrogen fixation reverse reaction. This is where we release energy that the bacteria is able to use. So many chemoautotrophs live in extreme environments such as deep sea vents, where they're able to utilize hydrogen sulfide. All right, again, the carbon cycle here, as far as prokaryotes are concerned, movement of carbon is going to be via carbon dioxide for the most part. We remove it from the atmosphere by the land plants and marine prokaryotes, and we return it to the atmosphere via the respiration of our chemoorganotropic organisms. The largest carbon reservoir in terrestrial ecosystems is within rocks and sediments, all right? And that carbon that is stayed within rocks and sediments is not readily available. We see a large amount of carbon available in land plants because of their production of carbon dioxide, all right? We use carbon dioxide from the air to synthesize carbon compounds here. We also see that a significant source of carbon compounds is humans, all right? We get this because of consumption of animal products, right? Consumers, such as our animals, use organic compounds generated by the producers and release carbon dioxide. Our most important contributor of carbon dioxide to the atmosphere is the microbial decomposition of dead materials. Right, so at the start, we have carbon that's going to be obtained from somewhere. If we get it, right, and we turn it for a photoautotroph, we're going to get energy from light, and we get carbon from inorganic materials or inorganic places. If we have a photoheterotroph, we get energy from light, and we are going to get our carbon from organic sources. If we have a chemoheterotroph, right, energy is going to be from inorganic oxidation. If we have a heterotroph, energy is from inorganic oxidation, right? If we have an autotroph or a heterotroph, depends on if we get from ourselves or if we get it from organic molecules outside of our own abilities. So here's a basic overview graphic of the carbon cycle. Get carbon from a lot of different places, right? Carbon dioxide is constantly floating around in the atmosphere. It's being released by respiration from animals and human emissions, things like that. It is taken back in through things like terrestrial photosynthesis. All right, and we see it also coming into the soil from weathering of terrestrial rocks. It's going to be leached off and also run off into creeks, rivers, things like that. Also, we see that we have marine respiration, which is going to cause carbon dioxide release, and we have marine photosynthesis, which is going to be the opposite of that. Main carbon source here we see is going to be stored within the soil. Also, at the deep levels of the soil, we have fossil carbons. And in the ground, we do have microbes and small prokaryotic organisms that are able to respire and decompose. For the nitrogen cycle, nitrogen is going to be a macronutrient as far as this is concerned in many organic compounds. So it's going to be recycled from organic compounds to ammonia, ammonium, nitrate, nitrite, and nitrogen gas. The largest pool of nitrogen is going to be in the gaseous nitrogen from the air. So this nitrogen is not usable by plants. So we have to have fixation of this nitrogen in order for it to be used. So that gaseous nitrogen is going to be fixed by the bacteria in the soil. Ammonia, however, is able to be used by plants or converted to other forms. We get ammonia from ammonification. This is where we have ammonia released during decomposition of nitrogen compounds. Ammonia released to the atmosphere only represents about 15% of the total nitrogen. The rest is going to be atmospheric nitrogen. Nitrification is the process where we convert ammonia to nitrate and nitrite. So in nitrification processes, such as in the soil, they're carrying that out by bacteria. Bacteria are going to perform the reverse process, so the reduction of nitrate from the soils to gaseous compounds, such as nitric oxide, is a process called denitrification. So we have nitrogen from the atmosphere, goes down into the soil. Nitrogen fixing bacteria in the root nodules of plants are able to get that nitrogen and fix it for that. The nitrogen fixing soil bacteria are going to convert it to ammonium. We also have nitrifying bacteria that are going to cause nitrite and nitrate formation. Decomposers such as funguses, mushrooms, things like that are able to take plant and organic material and decompose that and turn it into ammonium upon decomposition. All right, main cellular difference Unlike our prokaryotes, eukaryotic cells are able to compartmentalize various metabolic processes in different organelles. So the breakdown of certain food molecules provide energy takes place in the mitochondria, whereas photosynthesis takes place in the chloroplast. So compartmentalization of metabolic process makes the eukaryotic cells very efficient. All right, everyone. Thank you for watching the Cellular Metabolism Lecture. Please like and subscribe to the channel and visit our Patreon, patreon.com/twistedscience for more videos. I will see you all in our next video. Take care.